RuBPCase15-bisphosphate carboxylase, oxygenase, commonly known by the abbreviations RuBisCO, RuBPCase, or RuBPco, is an enzyme involved in the first major step of carbon fixation, a process by which atmospheric carbon dioxide is converted by plants and other photosynthetic organisms to energy-rich molecules such as glucose. In chemical terms, it catalyzes the carboxylation of RuBPCase15-bisphosphate, also known as RuBP. It is probably the most abundant enzyme on Earth. Versus alternative carbon fixation pathways. RuBisCO is important biologically because it catalyzes the primary chemical reaction by which inorganic carbon enters the organic biosphere, while many autotrophic bacteria and archaea fix carbon via the reductive acetyl-CoA pathway, the 3-hydroxypropionate cycle, or the reverse Krebs cycle. These pathways are relatively smaller contributors to global carbon fixation than that catalyzed by RuBisCO. Phosphenol pyruvate carboxylase, unlike RuBisCO, only temporarily fixes carbon, reflecting its importance. RuBisCO is the most abundant protein in leaves, accounting for 50% of soluble leaf protein in C3 plants, 20 to 30% of total leaf nitrogen, and 30% of soluble leaf protein in C4 plants, 5 to 9% of total leaf nitrogen. Given its important role in the biosphere, the genetic engineering of RuBisCO in crops is of continuing interest. See below. Structure. In plants, algae, cyanobacteria, and phototrophic and chemoautotrophic proteobacteria. The enzyme usually consists of two types of protein subunit, called the large chain, L, about 55,000 Daltons, and the small chain, S, about 13,000 Daltons. The large chain gene, RBCL, is encoded by the chloroplast DNA in plants. There are typically several related small chain genes in the nucleus of plant cells, and the small chains are imported to the stromal compartment of chloroplasts from the cytosol by crossing the outer chloroplast membrane. The enzymatically active substrate, ribulose 15-bisphosphate, binding sites are located in the large chains that form dimers as shown in Figure 1. Above, right, in which amino acids from each large chain contribute to the binding sites. A total of 8 large chains equals 4 dimers, and 8 small chains assemble into a larger complex of about 540,000 Daltons. In some proteobacteria and dinoflagellates, enzymes consisting of only large subunits have been found. Magnesium ions, Mg2+, are needed for enzymatic activity. Correct positioning of Mg2+ in the active site of the enzyme involves addition of an activating carbon dioxide molecule, CO2, to a lysine in the active site, forming a carbamate. Formation of the carbamate is favored by an alkaline pH. The pH and the concentration of magnesium ions in the fluid compartment. In plants, the stroma of the chloroplast increases in the light. The role of changing pH and magnesium ion levels in the regulation of RuBisCO enzyme activity is discussed below. Enzymatic activity, as shown in Figure 2. Left. RuBisCO is one of many enzymes in the Calvin cycle. Substrates. During carbon fixation, the substrate molecules for RuBisCO are RuBulose15-bisphosphate and carbon dioxide, distinct from the activating carbon dioxide. RuBisCO also catalyzes a reaction between RuBulose15-bisphosphate and molecular oxygen, O2, instead of carbon dioxide, CO2, products. When carbon dioxide is the substrate, The product of the carboxylase reaction is a highly unstable 6-carbon phosphorylated intermediate known as 3-keto-2-CARBOXYARABINITOL-15-bisphosphate, which decays virtually instantaneously into two molecules of GLYCERATE3-phosphate. The extremely unstable molecule created by the initial carboxylation was unknown until 1988, when it was isolated. The 3-phosphoglycerate can be used to produce larger molecules such as glucose. Also, RuBisCO side activities can lead to useless or inhibitory byproducts. One such product is XYLULOSE15-bisphosphate, which inhibits RuBisCO activity when molecular oxygen is the substrate. The products of the oxygenase reaction are phosphoglycolate and 3-phosphoglycerate. Phosphoglycolate is recycled through a sequence of reactions called photorespiration, which involves enzymes and cytochromes located in the mitochondria and peroxisomes. This is a case of metabolite repair. In this process, two molecules of phosphoglycolate are converted to one molecule of carbon dioxide and one molecule of 3-phosphoglycerate, which can re-enter the Calvin cycle. Some of the phosphoglycolate entering this pathway can be retained by plants to produce other molecules such as glycine, at ambient levels of carbon dioxide and oxygen. The ratio of the reactions is about 4:1, which results in a net carbon dioxide fixation of only 3.5. Thus, the inability of the enzyme to prevent the reaction with oxygen greatly reduces the photosynthetic capacity of many plants. Some plants, many algae, and photosynthetic bacteria have overcome this limitation by devising means to increase the concentration of carbon dioxide around the enzyme, including C4-carbon fixation, carboxylation, acid metabolism, and the use of pyrenoid rate of enzymatic activity. Some enzymes can carry out thousands of chemical reactions each second. However, RuBisCO is slow, being able to fix only 3 to 10 carbon dioxide molecules each second per molecule of enzyme. The reaction catalyzed by RuBisCO is, thus, the primary rate-limiting factor of the Calvin cycle during the day. Nevertheless, under most conditions, and when light is not otherwise limiting photosynthesis, the speed of RuBisCO responds positively to increasing carbon dioxide concentration. However, our descriptive knowledge will become more usable when we can translate them into quantitative models that can enable us to calculate the outcome of the reaction under a given condition. Since RuBisCO reacts with RuBP, ribulose 15-bisphosphate, first produces NADOL and next with CO2 that after some intermediate changes produces PGA, 3-phosphoglycerate. A biochemical model is developed to represent the effects of these steps quantitatively. Since carboxylation or fixation of CO2 is possible only after the synthesis of NADOL, thus it is suggested that the role of RuBisCO is to produce NADOL that is carboxylase and oxygenase, ENCO. Accordingly, RuBisCO is called an olase phosphoglycerase, EPGase, since it is neither carboxylase nor oxygenase. Regulation of its enzymatic activity. RuBisCO is usually only active during the day as ribulose 15-bisphosphate is not regenerated in the dark. This is due to the regulation of several other enzymes in the Calvin cycle. In addition, the activity of RuBisCO is coordinated with that of the other enzymes of the Calvin cycle in several ways. Regulation by ions. Upon illumination of the chloroplasts, the pH of the stroma rises from 7.0 to 8.0 because of the proton-hydrogen ion H+ gradient created across the thylakoid membrane. At the same time, magnesium ions Mg2+, move out of the thylakoids. increasing the concentration of magnesium in the stroma of the chloroplasts. RuBisCO has a high optimal pH, can be greater than 9.0, depending on the magnesium ion concentration, and, thus, becomes activated by the addition of carbon dioxide and magnesium to the active sites as described above. Regulation by RuBisCO activars in plants and some algae. Another enzyme, RuBisCO activars, is required to allow the rapid formation of the critical carbamate in the active site of RuBisCO. RuBisCO activars is required because the ribulose 15-bisphosphate RuBP substrate binds more strongly to the active sites, locking the carbamate and markedly slows down the activation process. In the light, RuBisCO activars promotes the release of the inhibitory, or, in some views, storage RuBP from the catalytic sites. Activars is also required in some plants, e.g., tobacco and many beans, because, in darkness, RuBisCO is inhibited, or protected from hydrolysis, by a competitive inhibitor synthesized by these plants, a substrate analog to carboxy-dearabitol 1-phosphate, CA1P. CA1P binds tightly to the active site of carbamylated RuBisCO and inhibits catalytic activity. In the light, RuBisCO activars also promotes the release of CA1P from the catalytic sites. After the CA1P is released from RuBisCO, it is rapidly converted to a non-inhibitory form by a light-activated CA1P phosphatase. Finally, once every several hundred reactions, the normal reactions with carbon dioxide or oxygen are not completed, and other inhibitory substrate analogues are formed in the active site. Once again, RuBisCO activars can promote the release of these analogues from the catalytic sites and maintain the enzyme in a catalytically active form. The properties of activars limit the photosynthetic potential of plants at high temperatures. CA1P has also been shown to keep RuBisCO in a conformation that is protected from proteolysis at high temperatures. RuBisCO activars aggregates and can no longer activate RuBisCO. This contributes to the decreased carboxylating capacity observed during heat stress. Regulation by ATP, ADP and stromal reduction, oxidation state through the activars. The removal of the inhibitory RuBP, CA1P, and the other inhibitory substrate analogues by activars requires the consumption of ATP. This reaction is inhibited by the presence of ADP, and, thus, activars activity depends on the ratio of these compounds in the chloroplast stroma. Furthermore, in most plants, the sensitivity of activars to the ratio of ATP-ADP is modified by the stromal reduction, oxidation, redox state through another small regulatory protein, the orodoxin. In this manner, the activity of activars and the activation state of RuBisCO can be modulated in response to light intensity and, thus, the rate of formation of the ribulose 15-bisphosphate substrate. Regulation by phosphate. In cyanobacteria, inorganic phosphate, Pi, participates in the coordinated regulation of photosynthesis. Pi binds to the RuBisCO active site and to another site on the large chain where it can influence transitions between activated and less active conformations of the enzyme. Activation of bacterial RuBisCO might be particularly sensitive to Pi levels, which can act in the same way as RuBisCO activars in higher plants. Regulation by carbon dioxide. Since carbon dioxide and oxygen compete at the active site of RuBisCO, carbon fixation by RuBisCO can be enhanced by increasing the carbon dioxide level in the compartment containing RuBisCO, chloroplast stroma, several times during the evolution of plants. Mechanisms have evolved for increasing the level of carbon dioxide in the stroma. CC4 carbon fixation. The use of oxygen as a substrate appears to be a puzzling process, since it seems to throw away captured energy. However, it may be a mechanism for preventing overload during periods of high light flux. This weakness in the enzyme is the cause of photorespiration, such that healthy leaves in bright light may have zero net carbon fixation when the ratio of O2 to CO2 reaches a threshold at which oxygen is fixed instead of carbon. This phenomenon is primarily temperature dependent. High temperature decreases the concentration of CO2 dissolved in the moisture in the leaf tissues. This phenomenon is also related to water stress. Since plant leaves are evaporatively cooled, limited water causes high leaf temperatures. C4 plants use the enzyme carboxylase initially, which has a higher affinity for CO2. The process first makes a 4-carbon intermediate compound, which is shuttled into a site of C3 photosynthesis, then decarboxylated, releasing CO2 to boost the concentration of CO2. Hence the name C4 plants. Carboxylation acid metabolism. CAM. Plants keep their stomata, on the underside of the leaf, closed during the day, which conserves water but prevents the light-independent reactions A, K, A, the Calvin cycle, from taking place. Since these reactions require CO2 to pass by gas exchange through these openings, evaporation through the upper side of a leaf is prevented by a layer of wax. Genetic engineering. Since RuBisCO is often rate-limiting for photosynthesis in plants, it may be possible to improve photosynthetic efficiency by modifying RuBisCO genes in plants to increase catalytic activity and/or decrease oxygenation rates. This could improve biosequestration of CO2 and be an important climate change strategy. Approaches under investigation include transferring RuBisCO genes from one organism in another organism, increasing the level of expression of RuBisCO subunits, expressing RuBisCO small chains from the chloroplast DNA, and altering RuBisCO genes to increase specificity for carbon dioxide or otherwise increase the rate of carbon fixation. One avenue is to introduce RuBisCO variants with naturally high specificity values such as the ones from the red alga Galdiaria partita into plants. This may improve the photosynthetic efficiency of crop plants, although possible negative impacts have yet to be studied. Advances in this area include the replacement of the tobacco enzyme with that of the purple photosynthetic bacterium Rhodospirillum rubrum. In 2014, 2 transblastomic tobacco lines with functional RuBisCO from the cyanobacterium Synechococcus elongatus PCC 7942, She 7942, were created by replacing the RuBisCO with the large and small subunit genes of the She 7942 enzyme, in combination with either the corresponding She 7942 assembly chaperone, RBCX, or an internal carboxysomal protein, CCMM35. Both mutants had increased CO2 fixation rates when measured as carbon molecules per RuBisCO. However, the mutant plants grew more slowly than wild type. A recent theory explores the trade-off between the relative specificity, i.e., ability to favor CO2 fixation over O2 incorporation, which leads to the energy-wasteful process of photorespiration, and the rate at which product is formed. The authors conclude that RuBisCO may actually have evolved to reach a point of near perfection in many plants, with widely varying substrate availabilities and environmental conditions, reaching a compromise between specificity and reaction rate. Since photosynthesis is the single most effective natural regulator of carbon dioxide in the Earth's atmosphere, A biochemical model of RuBisCO reaction is used as the core module of climate change models. Thus, a correct model of this reaction is essential to the basic understanding of the relations and interactions of environmental models. A new theory and model of the biochemical reaction of photosynthesis and the drawbacks of today's most widely used model of photosynthesis is discussed in Volume 29 of Advances in Photosynthesis and Respiration. Phylogenetic studies. The chloroplast gene RBCL, which codes for the large subunit of RuBisCO, has been widely used as an appropriate locus for analysis of phylogenetics in plant taxonomy. History of the term. The term RuBisCO was coined humorously in 1979 by David Eisenberg at a seminar honoring the retirement of the early prominent RuBisCO researcher, Sam Wildman, and also alluded to the snack food trade name Nabisco in reference to Wildman's attempts to create an edible protein supplement from tobacco leaves. See also Bibliography. See here for the mechanism of the RuBisCO catalyzed reaction. RuBisCO. RCSBPDB molecule of the month. The Plant Kingdom's Sloth. Protein Spotlight article on the slothful enzyme RuBisCO.